Welcome to Bison Jam, the podcast that covers wide-ranging topics from Yellowstone National Park and the greater Yellowstone ecosystem. I'm your host, Hugo Sindelar, an assistant professor of film at Montana State University. Today, we're continuing our series about the hot side of Yellowstone, where we explore the thermal features in the park. I am again joined today by Dr. Dana Skorupa, assistant research professor in chemical and biological engineering here at Montana State University. In this episode, we're continuing our discussion of bioprospecting by focusing on discoveries that have been made in Yellowstone. Welcome back, Dana.
Thanks for having me again.
So, Dana, one Yellowstone bioprospecting story that's perhaps the most famous is Taq polymerase. Can you kind of share the history there?
Sure. In the 1960s, researchers isolated a thermophilic bacterium, which they named Thermus aquaticus. And they isolated this organism from Mushroom Hot Spring in Yellowstone National Park. And so Thermus aquaticus had been grown and studied in the lab for a couple decades. But in 1985, scientists took an enzyme from this Thermus organism and used it to invent a new laboratory process called polymerase chain reaction, or as we more commonly call it, PCR, which is used to replicate DNA. And the enzyme discovered was named Taq polymerase because the T stands for thermus and the AQ stands for aquaticus, this organism from Yellowstone. And it really made PCR practical. So this Taq polymerase enzyme could withstand the heat of the PCR process without breaking down like a normal polymerase enzyme would. And this has been one of the most important enzymes to be discovered and used in the field of molecular biology.
And so what are some of the applications we see of PCR?
So thanks to Taq, PCR has become a cornerstone of modern science and medicine, with applications in everything from diagnosing diseases, solving crimes through forensic analyses, tracking pandemics, like in our recent world, to advancing genetic research and additional biotechnology applications. And I can't stress enough the important commercial value of this enzyme. So I looked up some numbers before our session today.
Cool.
And these numbers are from 1991, mind you, but Hoffman LaRoche acquired this Taq polymerase enzyme patent for $330 million back in 1991 and has reportedly earned up to $2 billion in royalties from this particular technology development. What's also really important to know is that companies that sell Taq polymerase enzymes can earn profits. But Yellowstone National Park, at the time of this development, has received no direct royalties or financial benefits. So I know earlier we talked about the benefits sharing agreement that researchers enter into when they want to conduct research in the park. But at the time when Thermus aquaticus was first isolated from Yellowstone and grown in laboratories, there was no such benefit sharing agreement. And so that is why in 2010, the National Park Service enacted or required benefit sharing agreements so that the park would retain a reasonable share of the profits when park-based resources yield something of commercial value, like Taq polymerase.
So you said it sold for $330 million?
In 1991.
In 1991.
Yeah.
Wow. And the park's never seen a dollar from any of that.
Exactly, yeah.
So do we know some of the history of, why it was Dr. Thomas Brock, correct? Do we know why he got started in Yellowstone, or?
He was one of the original microbiologist pioneers in Yellowstone and sampled a whole slew of different geothermal basins and springs throughout Yellowstone National Park. Again, like I said in our previous episode, Thermus aquaticus is kind of like a weed microorganism. So it might have been that.
Does that mean it's everywhere when you refer to it as a weed? Or?
No, it has certain chemical conditions, temperatures, pHs that it prefers to grow in. But it would be a pretty common microbe to be able to isolate and grow maybe more easily in the lab. So whether that was why Brock had this established culture so quickly and had been studying it for decades, I'm just guessing at this point. But for us, it's a common, easy to grow thermophile that we run into a lot when we're growing thermophiles in the lab.
And then, so that enzyme is useful in PCR or polymerase chain reaction, right?
Yeah.
Because it's thermostable during that process?
Yes. So unlike, say, we have polymerase enzymes in our own human cells, right, that are replicating our DNA. But part of the polymerase chain reaction process requires heating up that molecular mixture at 72 degrees Celsius, and in some cases up to 95 degrees Celsius. So you've got to have an enzyme that's stable up at those high temperatures. And polymerase enzymes from more what we call mesophilic organisms like you and I would fall apart or degrade under those high temperature conditions. But the enzyme from Thermus aquaticus stays intact and performs at a high rate and accurately just fine at those high temp conditions.
And what is can you kind of, for someone out there who's a non-scientist, what essentially does PCR do?
It's photocopying a piece of DNA. And in most cases, you're photocopying specific subsections of the genetic material. So along with that polymerase enzyme, you also are including these little short synthetic pieces of DNA that specifically tell that photocopying process where to start and where to stop. And what's amazing about the PCR process is that through those little synthetic pieces of DNA that you include and that polymerase enzyme and a couple other additional reagents, you could start from a single piece of DNA and photocopy that to billions of copies in a matter of 35, 40 minutes. So you can create enormous quantities of these DNA copies in a very short amount of time, but in an accurate, reliable way.
And people out there may have heard of PCR in relation to the pandemic we just went through, COVID-19, one of the tests that was deemed more reliable, right? There were different tests for whether you had COVID-19. It was PCR testing.
Right, yeah.
So can you talk about what that process is doing to test if someone has an infectious disease?
Sure. So like you just described, we used rapid-based PCR tests to diagnose COVID-19 infections, right? And what we're doing is essentially detecting a very small amount of the viral genetic material from this coronavirus, from, say, a nasal swab, and then photocopying the coronavirus genetic material using that PCR. And because TAC enzymes can rapidly and reliably amplify that viral genetic material, it allows healthcare providers and scientists to identify infections from even very small quantities of original viral material, right? And if you think about it, without that rapid-based PCR, large-scale testing, early detection, which were really essential tools for tracking and controlling the spread of COVID-19, would not have been possible.
And that testing is used for all sorts of infectious diseases, correct?
Yes.
So it's used across the spectrum to kind of help us determine if you have caught some sort of infection. So it's been really revolutionary for medical science.
Yes, that, and even our own. We think about the 23andMe, like genetic testing that we'll do, that lots of people are now doing across the world. All of that originates from the development of that TAC polymerase enzyme.
And PCR and TAC polymerase play another role for our true crime listeners out there, right? It's big in forensic science, from what I understand.
Yes, and identifying genetic material from at crime scenes and from either victims or potential perpetrators, is really key in linking potential perpetrators to, say, specific crime locations. Yeah.
And so you're kind of talking about how this technology has been developed, right? So this one company that purchases in 1991, anytime you do PCR, they get a share of that profit? So I'm curious how that works.
I would say there are now a lot of spin-off variants on TAC polymerase enzyme, and that it's been genetically engineered and modified. So it's not just from this isolate from Tom Brock at Mushroom Hot Spring in the 1960s. Okay. There's lots of now different variants, other thermostable versions of the Taq polymerase enzyme that are isolated from other thermophiles and then have been genetically engineered so that they are stable for longer periods of time, say at high temperature, or that they make fewer mutations or mistakes, right? Your typical run-of-the-mill Taq polymerase enzyme makes an error 1 out of every 1,000 nucleotides that it's incorporating when it makes a photocopy of the DNA, which isn't desirable, right? I mean, that's what drives mutations and evolution through living organisms on all scales across our planet, but it's not desirable in these particular molecular biology applications. So we've genetically engineered it to make fewer mistakes. Maybe it only makes an error 1 every 10,000 nucleotide base pairs that it's incorporating. Those types of things. So we've tweaked it and modified it, and there are now probably lots of variants sold by lots of different molecular vendors across the world. Mm-hmm.
So we talked about kind of the role of PCR and Taq polymerase in infectious diseases, also forensics. I'm curious, are there any other applications?
I mean, we use them in thermal biology to help study microorganisms that we can't grow in the lab, right? So I've talked quite a bit about how my goal is to try and culture new, novel thermophiles that have these unique metabolisms that we can tap into different biotechnology applications. But we estimate of all the microbial diversity on our planet that we can grow in the lab, it's now like 0.01 or 0.1% of all the microbial biodiversity on our planet. So if we can't grow them, one way that we can kind of get around and still be able to study these organisms that on a global scale are conducting really important functions on our planet is by studying their DNA. And without having to grow them, what we call culture-independent methods, we can extract out their DNA, photocopy it, and sequence it. And part of that sequencing process and the photocopying in the lead up to sequencing environmental DNA uses polymerase enzymes. Okay? And that allows that once we've sequenced all of this genetic information from different environments, whether it be ocean or hot spring, we can now start to sequence the genomes of these microorganisms, assemble them, and start to ask questions like, what are they doing? How are they doing it? And how could we then apply them to different technology applications that might benefit society in some way? So it really has revolutionized for microbiologists our field of study as well, getting around this roadblock of not being able to culture organisms and study them in the lab in that way.
And do you do any culture independent work yourself?
Yes, absolutely. Mm-hmm.
And so have you had any promising results from that in the work you've done?
I don't do whole genome assembly.
Okay.
Like Dr. Bill Inskeep. Dr. Bill Inskeep has discovered, I think, 2 different phyla of archaea from hot springs in Yellowstone through this process of DNA, whole genome sequencing and genome assembly.
And that's called metagenomics?
Metagenomics, yes.
Yes. And so we discussed that in a previous episode with it. Yeah.
So there are absolutely researchers here at Montana State that have, discovered whole new taxa of different groups of bacteria and archaea using this process of DNA sequencing and metagenomics. Yeah.
Well, let's transition from kind of, Dr. Inskeep's work and some of the Taq polymerase stuff into your work. So you've done some bioprospecting. We kind of talked in our previous episode about that process. But in this episode, we want to kind of jump into what you found and some of the findings. And I think a lot of the work you've done has been looking at thermophiles that might help break down plastics, right? And so the curiosity that I have is, why would you go to Yellowstone to look for a microbe that breaks down plastic when there isn't a lot of plastic in that environment?
Yeah, that's a great question. Well, my answer would be every living organism needs access to the building block of life, which is carbon. And it turns out plants are an easy to use source of carbon for many microorganisms, including hot spring microbes and those thermophiles. So if you ever get a chance to go to Yellowstone, take a look around at geothermal features that you're seeing from the boardwalk and notice all of the downed trees, pine needles, and plant biomass that are kind of either floating on the surface or along the bottom of these thermal pools. So microbes take advantage of that plant material that's around in their environment. For thermophiles, some species express or produce enzymes that help them access the carbon that's tied up in that plant material. And it just so happens a lot of the enzymes that a microbe would use to break down plant material are they're not specific. They're perfectly happy chewing on plant material to access carbon as they would the carbon that's tied up in a plastic film like your plastic water bottle or single-use plastic bag. So it's kind of like the microbe doesn't intentionally, in some cases, produce that enzyme for that purpose.
Okay.
It's using it non-specifically. It's like, oh, plant biomass isn't around, but I've got this plastic material. Sure, I'll try and chew on that plastic and draw out the carbon molecules and incorporate it into my biomass for growth and reproduction.
How did you get started researching the breakdown of plastics? I'm curious.
There's been a strong focus in the Peyton Lab on trying to find microbes or thermophiles in particular with different biotechnology applications. And in I would say the mid 2010s, there was a real focus on trying to, from government, from industry, trying to come up with ways of recycling more types of plastic material, right? Right now in the United States and across the globe, we only recycle plastics that are described as types 1 and 2, right? But if we look at the bottom of a lot of the plastic materials that we consume and use, they fall in that category of being a type 3 through 7, which we have no means of recycling. So I think there's a real push to try and come up with more green technologies to recycle more plastic materials. We kind of jumped on that bandwagon. And there are a number of advantages that thermophiles hold for finding microbial-based ways of recycling plastics and then thinking about upcycling them into some new value-added product.
It's clearly important to recycle plastic. So maybe we can start with, what does plastic recycling look like now?
Yes. So currently we can recycle plastic types that are described as types 1 or 2. And primarily we recycle them into products that are being reincorporated into textiles primarily, but also some playground equipment and things like that. The other types of plastic materials, 3 through 7, we have no means of recycling, right? So when you go to your local recycling facility, it's asking you to sort types 1 through 7 separately. Types 1 and 2 are being recycled. 3 through 7 are being landfilled. Whether we know that or not, it's kind of more getting you in the practice of sorting plastics so that hopefully one day we have a means of recycling them. So there has been work looking at the use of microorganisms to recycle plastic materials. Okay. The thing we should know about coming up with plastic recycling streams is that it's not just a matter of breaking down those plastic materials. We have to have some economy for what we can upcycle them into. So for plastics type 1 and 2, we're primarily upcycling them into textiles, playground equipment, some kind of minor industries like that. Textiles are the big one.
Okay.
So there have been breakthroughs in trying to find microbes that break down these 3 through 7 plastic wastes. A team of researchers out of, I think it was Japan or China, isolated a bacterium from a plastic recycling facility that could literally eat plastic as its food. And really interestingly, this organism had actually evolved a specific enzyme, the PETase enzyme, to break down plastics. So that means the plethora of plastic waste that are around in our everyday world in just the last 80 years has caused an evolution in these microbes to be able to evolve specific enzymes to degrade plastics. And additional research teams have also worked on these enzymes from this original bacterial organism and engineered it so that it could catalyze this plastic degradation reaction 3 times faster than what it would typically do. So there's lots of now genetic engineering to try and improve the performance of these enzymes. Interestingly, I found a study also that showed that this same enzyme has been genetically transformed into a fast-growing bacterium that lives in marine environments. And this engineered strain has been shown to break down microplastics in saltwater environments. So trying to offer a solution to this marine plastic pollution problem. What leads us to thermophiles, though, from all of that, is that every plastic material has what's called, I'm going to get a little bit off topic. I promise I'll bring us back.
That's okay.
Every plastic material has what's called a glass transition temperature. So at low temperatures, plastics are really rigid and firm. Like that's what makes them such a great packaging material, right? But above, as we heat up that plastic material, above that glass transition temperature is when the plastic becomes more malleable.
Okay.
And when it's more flexible. And that's when these biological enzymes can really burrow into the material and start pulling out those carbon molecules and basically eating it as their carbon and energy source. So the rub is that for most plastic materials, that transition temperature where it reaches this flexible, malleable state is really high, say on the order of 140 to 175 degrees Fahrenheit. So that means for microbes to be effective, to actually be used widespread in recycling practices, the enzymes they produce really need to be thermostable, okay? So we have yet, or the research work that myself and my honors college students do is geared towards that, of trying to find plastic-eating microbes that produce these heat-stable enzymes. And we think Yellowstone and its thermal environments are a perfect place to try and bioprospect and find that.
140 would still be pretty hot for Yellowstone, right?
Yes. Yeah. And so I will say...
And we're talking degrees Celsius,
yes.
Right?
So over the years, our research work has evolved. And we found through our research that it's really difficult for microbes to chew on those plastic polymers that are in kind of that parent form, in that as a film, or what you would think of as being like a plastic bottle or a plastic bag. So we now do what's called pre-treatment. So we partially break down the plastic before we feed it to our hot spring microorganisms. So that improves their activity and their efficacy. In the process of pre-treating it, I should say this is done with collaborators in the chemical and biological engineering department here at MSU, Dr. David Hodge.
Okay.
This helps to break down the plastic material into these soluble forms and these much shorter carbon chains that are much easier for microbes and biological enzymes to start kind of chewing on. The way I like to picture it or describe it to my students is kind of like these enzymes are like little Pac-Mans that are chewing on the ends of the chain. And it's a lot harder if those chains are tens of thousands or hundreds of thousands of carbons in length and these really complex amorphous structures. So the pretreatment process chops them up, linearizes them, and makes them more amenable to biological degradation. And they're also poised at lower temperatures then.
So as you've been talking about your work and kind of the breakdown of plastics in general, a question that I've had is, what do you mean by breakdown of plastic, right? Because if you landfilled something, someone's going to say, well, eventually, probably over thousands of years, right, or at least hundreds of years, it'll break down. Mm-hmm. But what do you mean in terms of this research, in terms of what do we get in the end when we break it down?
Great. So it is more on the, we think of a typical plastic water bottle as naturally biodegrading on the order of like every 500 to 1,000 years. So it's really slow natural degradation rates. When we're talking about microbes breaking down this, what they're really doing is pulling out those carbon molecules, taking it up into their cell, and using that to grow and divide on. Okay, so so just like you and I are noshing on our fruits and veggies and
Right.
food sources, that the carbon in that plastic is their food source. So they're growing on it to create more biomass. And so it's kind of a two-pronged approach of both using the microbes to break down, consume that plastic material. But once you have microbes growing on that carbon and energy source in large quantities, now you want those to use those microbes for some new product, right?
Right.
So there are microbes that will make bioplastics as a storage compound.
Okay.
So whereas you and I store our excess energy as fats, some microbes will produce these storage compounds that are actually plastic-like compounds. So we could both use microbes to break down plastic waste that we've already accrued on our planet and make new plastic products from that.
And would those bioplastics be able to make like a plastic water bottle? Would they make textiles? What would they be used in? Or we don't know yet.
I don't know.
Okay.
I don't know what specific like target products are being made. They're polyhydroxybutirates and polyhydroxyalkanates are the two types of bioplastics that microbes make. But like what ultimate shelf product, you would see, I don't know offhand. We also think of these microbes maybe being able to make other useful products too. So if we grow up their biomass, maybe they could make different chemical precursors, oils, lubricants, different other value-added products. So our whole system is trying to be circular of, okay, they can both degrade down the plastic material, but what new thing that has an economic value and an economy can they transform that degraded material into?
What do you think, and this may be some conjecture on your part, but I'm just curious if this research was to upscale to, say, a level of Taq polymerase, right? It really becomes quite prevalent. What does that look like in terms of a plastic pipeline? So I used to work in kind of water treatment, right? And so that would be, you might take in a wastewater stream and then you have water that meets a certain standard, clean water on the outflow. Is that, we all put our plastics in our recycling bin, it gets brought to some area where it's processed in these bioreactors. I'm just curious, what is you recycle your plastic water bottle to new plastic product. What does it look like in between if these technologies mature like we hope?
Yeah. I think the ideal goal is for 3 through 7 plastics that you don't have to sort them. You just dump it all in a collective bin, right? And then you're taking those plastic water bottles, products, styrofoam, whatever it may be, heating them up in some kind of stainless steel reactor to high temperature where it partially breaks them down, shortens those carbon chains, makes them dissolve in the liquid water solution that we've put them in. And then you feed them into a bioreactor with maybe a consortia of different high temperature adapted microbes. It's advantageous if they're thermophiles. One, because then you don't have to cool down that reactor with those kind of now dissolved plastic materials, right? You can keep it at high temperature because the thermophiles like that. And maybe there's a consortia, a mixture of different thermophiles where one attacks one particular plastic material, another attacks another. And ideally, you have what's called a consolidated bioprocess where along with uptaking the plastic materials and using it as carbon and energy source to make biomass, then they're also producing some new bioproduct. But maybe we also recognize it might be more complex and that maybe one group of thermophiles is doing the degradation part and you've got another group of organisms, maybe they're thermophiles, maybe they're normal mesophilic microbes that are producing the new value-added product. So it might be kind of a combination of the two.
Cool. I want to step back. So we've talked a lot about this process kind of as what we hope in the future and what's been done. What are you specifically finding in your research?
I think I can tell you what we're finding is a little bit limiting.
Okay.
I think we've had much more success over the years now that we're growing these thermophiles on these kind of partially broken down plastic materials. They prefer to grow where that plastic carbon chains dissolved in their liquid microbiological media, where it's shorter and they can attack the carbon chains easier, having a lot more success with those types of material or waste types. It's still limiting with respect to how much they grow on it. We never really see high numbers. So whether it leads to industrial scale, I'm not quite sure it will get there. But what if we have a thermophile that's growing, we find the enzymes that are doing it, we clone them into some other E. coli growing organism that can do it really fast. There's I think the advances in genetic engineering now lend itself to
taking the technology further.
Mm-hmm. We even have collaborators that we're starting to work with who want to use AI technology to make predictions about what would be the idealized chain length, form, side chain groups, functional groups that you would add on to those plastic molecules that would make it the ideal biodegradable compound for a microbe to uptake and consume. So there's some cool collaborations that are starting in that realm too, using AI.
So still very much an open area of research.
Mm-hmm, definitely. And I think oftentimes it's really still driven by how cheap it is to make new plastic materials. And so it's still just industry-wise, unless we force industry to come up with ways of recycling and using more recycled plastic materials, it's still just so much cheaper to use virgin plastic materials and make new product using then petroleum-based support. So.
Are you going to keep looking for new thermophiles in Yellowstone? How long do you foresee this research going?
Yeah, I think in recent years we've kind of switched gears more to like plant biomass degrading.
Okay.
Which I think we're going to talk about in a little bit. So it does kind of ebb and flow based on what grant proposal crawls are out there and where the interest is at the federal level, and it has waned a little bit in recent years, that interest in that area of coming up with recycling streams. Yeah.
So we can go ahead and jump into the plant biomass now. So that, I think, relates to bioethanol, right? It would be fuel for vehicles or anything that takes gas.
Yes, correct. Yeah. So bioethanol is a type of renewable fuel that's made by fermenting sugars found in plants. It's primarily used as a substitute or additive to gasoline, right, in transportation fuels. And today, most of our bioethanol comes from corn. However, there's a lot of interest in using leftover plant materials from agriculture, from the forestry industry, or other industrial processes like food processing to reuse those plant waste materials, of which there's a lot of biomass from, as a sustainable resource in producing bioethanol, right? So there is a real concern we have with growing corn for fuel, and we create this kind of food versus fuel competition when we're growing corn for fuel production and not using it for food production, or if we're converting agricultural lands that used to produce a wide variety of crops to now just growing fields upon fields of corn for our transportation fuel needs. Yeah.
So you've done work in this area as well?
Yes, also with my Honors 275 students. So we're looking at trying to find thermophiles that could be incorporated into this plant biomass to fuel bioconversion system.
And any promising research yet?
Yeah, so I had students this past fall who they each student team kind of took on a different plant waste material. So I had one student team that was looking at corn wastes and corn stover, as we call it. One that was looking at the waste from food industry, and another one that was looking at the waste from just simple lawn clippings. And the team that was growing their thermophiles on these grass lawn clippings had some really great enrichments going of a whole diverse cohort or consortia of different types of thermophiles growing on that type of material as its sole carbon and energy source for growth. So that was really neat.
So I'm curious, I was curious with the plastics, what would this look like if this technology matured?
Yeah.
Someone would drop off their grass clippings from their lawn
Mhm.
somewhere, in the city they're in, and that would be turned into fuel. So if it went well, where would we end up?
Yeah. Okay. So, um, similar to these plastic wastes, plant biomass wastes are tough materials, but they're really rich in these different compounds that microbes like to utilize for their carbon and energy source. And so, the reason we need to use thermophiles in this process is that typically we have to pre-treat the plant waste materials. And that pre-treatment process often uses high temperature, extremes of pH, and those extreme conditions are needed to kind of loosen up that biomass structure and make it more accessible for microbes to degrade. So you could envision taking that grass clipping waste, pre-treating it under high temp with maybe some combination of an extreme of pH, and then exposing that loosened biomass structure to a thermophile without needing to cool down the system, right? You could expose it to your thermophile that then will start to degrade, again, the carbon molecules tied up in that material type and uptaking it and incorporating it into their biomass and growing on it.
And then they would produce the bioethanol?
Yes, or it's that thermophile breaks down that plant material into simple sugars, which then you feed into a fermenter and have some mesophile that takes the sugars and converts them into ethanol. So ideally, again, the ultimate goal is to find one organism, a consolidated bioprocess where it's one vat where you kind of put in the partially broken down plant material and you get out the bioethanol. But so far to date, the most promising systems have been these two-step process where the thermophile does the first step, breaks it down to simple sugars, and then a mesophile ferments it really quickly into bioethanol.
And mesophile, again, for those out there, is someone who's at a more moderate temperature, right? Under 70 degrees Celsius or 60?
I mean, under 40.
40, wow.
Yeah. Mm-hmm.
And so where do you stand today with some of this work?
Well, our fall 2024 class was the first time that we tried those particular substrates.
So recently.
Yeah. So I had some undergrads. So I should say oftentimes students who take the class and they continue to work in the lab. And so we got to the point at the end of spring semester where they had ID'd some other organisms through DNA sequencing, isolations, and now doing like just some initial growth curves to try and see like, okay, how fast can they actually grow on this plant waste material, these grass clippings, and what are the actual maybe enzymes that they're using in this process. Yeah.
So I think we can jump into, you have a story about a food company that started with thermophilic research in Yellowstone. So I'm curious if you can tell us that story.
Yes. So two researchers, Mark Kosabo and Rich Maker, who are both affiliated with Montana State University, they isolated heat-loving fungi and acid-loving, I should add, fungi from Norris Geyser Basin in Yellowstone National Park. This particular fungi is from a taxonomic group called Fusarium. And the researchers they nicknamed it FY or FY for short. And the FY stands for Fusarium of Yellowstone or FY. So keep that in mind. Interestingly, Mark and Rich originally studied this fungi because of the interesting renewable fuels that it made. However, and they had kind of like a startup company that they were trying to kind of create on campus in our biotech park. However, this fungi never really made these fuel products in industrially relevant quantities. Okay. But through their research, they found that this thermophilic fungi was nutritionally dense, that it produced high quantities of all the essential amino acids and proteins, and a whole heck of a lot of fiber. And so they got this kind of spark idea that pivoted their biotech research. And now this fungus from Yellowstone, Fusarium, is being used as a vegan-friendly protein source. So they used it in food in food products.
Okay.
So Mark and Rich started a food company called Nature's Find, where find is spelled F-Y for Fusarium
Yes.
of Yellowstone, N-D. And right now, the primary products that they make using this Yellowstone thermophile are yogurt and breakfast patties. And they're marketed as these kind of vegan-friendly food products. Now, Mark and Rich actually sold Nature's Find to some corporate partners out of Chicago, I believe. And I looked it up. The company's currently valued at $463 million from this biotech development. Yeah.
All from this fungi found in Yellowstone.
Yeah.
So is there any more to the story of this fungi?
Yeah. I guess I'd like to add that there are a number of advantages of growing protein in this way in comparison to where we traditionally get a lot of our protein in our human diet, right? Which is really from animals, right?
Yeah.
And cattle. So this particular organism, we can grow in cafeteria-sized lunch trays. Okay. So we can grow our food, our protein source, where people live. We can grow them in factories, in urban areas. We don't have to worry about the extraneous costs of shipping cattle right to a slaughter farm and then out to market and distribution across the country. They're also, because of this nutrient-dense proteins that they produce, in less than 4 days, a tray of these fungi develops the protein equivalent to 20 to 25 chickens. So very high protein content, right, for the amount of biomass that you're producing. It also, because it, in addition to liking high temperatures, likes acidic conditions, that means that when we grow it in these factories, we can grow it in a rather acidic liquid that's inhospitable to bacteria and other fungi and other organisms that might contaminate it. So we don't need to use a lot of antimicrobial compounds when we're growing this particular protein product. It also has low land use, okay, right? Because we're growing it in factory settings, it can be grown using 99% less land mass than we would need for beef. And it also has low water use needs. Like think about even if we are growing plant-based protein sources, right? That requires a lot of rainfall, irrigation, right, to provide those plants to be able to grow them to high quantities. So it has very low water-intensive practices, which make it ideal from a whole slew of different sustainability perspectives.
Do you know anything about how because they weren't looking for food initially, you said.
Yeah.
So how did they discover that this was a food source?
I think some of the analyses that they do when they're looking for these fuel products that they made, it also just kind of spits out some other information about other compounds that are being made, amino acids, fibers, different things like that. And so they could see maybe their quantities for their fuel products that maybe they were originally interested in were pretty low. But hey, geez, it makes a whole slew of amino acids and it makes these different variety of fibrous compounds and things like that. So yeah, sometimes it's the methods analysis that you're using spits out a whole lot of data, and maybe you're targeting one specific type of data analysis, but it gives you a lot of whole other information, and they really oftentimes we ignore it. But in the case of Mark and Rich, they're like, hey, wait, it also makes these other products that could be interesting.
And how did they discover that it was edible?
That I don't know. Yeah,
because
how do you
right,
go from? Yeah,
I mean, some mushrooms, right, you can get in a lot of trouble if you eat the wrong species of mushroom.
Totally. Yeah, that I don't know the answer to. It does when you see it in the lunch size trays. It has the consistency and texture of like chicken. Like it looks like
interesting
chicken. Yeah.
Have you eaten it?
No. But that's what I've been told is that it has this like chicken-like texture and appearance. Yeah.
Well, maybe we'll have to see if they're around for a future episode.
Yeah.
I think this also might have some applications to space exploration. Is that correct?
Yes. Yeah. So if we think about current diet for our astronauts, oftentimes they're eating freeze-dried foods, right? Those are kind of a staple of the space program. But there's a couple different problems with freeze-dried foods. One is that when we freeze-dry products, it reduces their nutritional quality. So maybe fewer protein, lower concentration of proteins, lower concentration of fibers, and it also shortens the shelf life of those particular food products. So as NASA hopefully gears up towards these more deeper missions into space and ultimately hopefully going to Mars, yeah, we've got to be able to feed our astronauts nutritional food that maintains viability and long shelf life for those longer distance missions. So, in contrast to that, folks at Nature's Find and researchers here at Montana State University, we're kind of promoting this fusarium organism, right? It can be grown in a small space, like I described. It can grow in those lunch-sized trays, and it can be grown repeatedly. So instead of sending astronauts into space with all the food that they would need for that entire 2-3 year mission, whatever it may be, we could grow fusarium while the space travel is happening from an initial microbial sample and regrowing these fungi repeatedly. We also tend to think that these fungi would be easy to grow with little active involvement from our astronauts.
Okay.
And again, it has that high nutritional quality to it. And interestingly, it can also be converted in a variety of different product types. So I know I told you it kind of has that chicken-like composition, but it could also be made into liquids, powdered forms. So you can kind of create a variety of more interesting food products using it than what they would traditionally be getting in their diet.
So all from looking for another application in Yellowstone, we developed this new food source.
Yeah, and a collaboration with Nature's Find and Montana State University researcher Ross Carlson sent up some of these fusarium on a SpaceX rocket to the International Space Station to test whether this fungi was tolerant to other extreme conditions like it would experience in space. So its tolerance to UV, radiation, microgravity conditions, things like that. And I believe they found that it did just fine.
Did the astronauts eat any of it yet?
That I don't know.
You don't know?
Yeah.
So are there any other potential biotech discoveries out there that you think?
Those are the two primary ones that the Taq polymerase, like that have come to commercial market. The two primary ones that I think of are Taq polymerase and these Nature's Find, like vegan-friendly food products that have come to a commercial market, at least. Mm-hmm.
So I think that speaks, these two technologies, biotechnologies that have come to commercial market, Nature's Find, Taq polymerase, speak to maybe the importance of basic research, right? And this idea of just going out in Yellowstone and looking for things. Do you have any, what do you think about the need to just study these thermophiles because we don't know what's out there?
Yes, absolutely. I think the two basic questions are knowing the answer to the questions, who's there, what are they doing, and how are they doing it? We have to be able to answer those fundamental questions about microbes from these natural environments before we can start to build on that and say, okay, how can we apply that to different technologies or benefit society in some way? So we have to have a solid fundamentals understanding of what metabolisms these microbes encode, what they could potentially doing, what they're actually doing in their environment, like activity-wise, before we can start to say like, oh, it does this really cool function that could be useful to this industry or in this environmental cleanup mechanism, et cetera. Yeah.
So people can think that there's uses out there that might just start as investigating these organisms, because people might be driving around listening to this, go, why should we study these little bacteria?
Yeah, the basic leads to the applied,
right.
And you need to put in the effort and interest into basic biology, and know that often spurs off these unique ideas of how to apply that information and use it to benefit society in some way.
So thinking about Nature's Find, we talked about how the park didn't get any royalties from Taq polymerase. Is that a different story now?
Yes, absolutely. I don't know what exact royalty percentage Yellowstone, the National Park Service, has gleaned from this Nature's Find commercial development, but absolutely, they're getting their cut.
So I kind of want to wrap up, Dana, with a couple questions. The first one is, what excites you most about thermophilic-based biotechnology as we look into the future?
Yeah. I think what excites me the most is their potential to transform how we do different things in our society, whether it be how we produce our energy, the types of materials that we make, and how we can incorporate this bio component into new materials, new products, and make these processes more sustainable and maybe even more efficient in the process. Um, I also think there's a huge component in something I is not an area that I conduct research in, but synthetic biology and how we're learning how to engineer microorganisms using these thermophilic enzymes to expand their metabolic capabilities, right? So taking an enzyme from a thermophile and cloning it into some organism that we already use in a commercial application to make it more functional at under certain extreme conditions, right? And this really opens the door to new industrial biotechnologies that are, again, cleaner, faster, less dependent on resources that we have on our planet that are quickly dwindling and running out. I'm thinking like of fossil fuels
Right.
in a lot of our applications that I do.
So, Dana, I'm curious, as someone who does research into thermophile-based biotechnologies, what's your favorite part of this research? Why do you do what you do?
I think it's really interesting just how applicable the use of thermophiles are in different technology applications. I mean, we've talked today about using them in fuel production, in food production, in breaking down different waste. We haven't even discussed cleaning up different contaminated environments and how we can use them in those technologies. So I think the plethora of ways that we could potentially use these organisms is vast. And that really excites me, right? I think there's always a utility and a use for these types of organisms. And these kind of burgeoning technology developments, remediation questions we have in our society, oftentimes we can find a thermophile that can help in informing a solution to whatever it is we're interested in.
Do you think some of these applications of microbes, this is just shooting off the cuff for you, but you see 20, 30, 40, 50 years down the road, do you think a lot of these microbial-based processes that you're researching will play a larger role in society?
Oh, definitely. Yeah, I think we have a whole unit at the Thermobiology Institute and Center for Biofilm Engineering where I work. We're discovering what we call engineered living materials, right? So we're finding different ways of using microbes to make biocement, make whole slews of different products. And we're just kind of scratching the surface of it. So yes, absolutely. And I think their ability to quickly and on a large scale, like produce certain products or degrade certain things is a real advantage to using microbes as opposed to other biological organisms. Yeah.
So that leads me to my last question. If you hoped listeners took away one thing from this podcast about thermophile-based biotechnology, what do you think they should know?
I would want people to take away that thermophiles are nature's extreme problem solvers and that they could be key in helping us build a more sustainable and innovative future through the different products they make or through the different functions that they can perform. And that these organisms and the powerful enzymes that they make might be key in developing the next like PCR-like innovation. And that they might also be the answer to help us turn what we usually think of as throwing away, right? An invaluable waste product into something that has an economic value, whether it be a fuel, a bioplastic, or a green chemical.
Well, Dana, I think that's a great place to end it. Thank you so much for joining us again on the podcast. It was great to have another conversation with you.
Thanks for having me.
And for our listeners, this was the last episode of our second season of Bison Jam. We appreciate you listening as we explored the hot side of Yellowstone. We have exciting things in the works for next season. And we hope you'll join us for season 3, coming in summer 2026. Thanks so much. Bison Jam is a production of Hugo Sindelar Media, LLC. I'm the director and producer, and Ashley LeBau is our editor and story consultant. This episode was also filmed by Ashley LeBau. Our guest was Dr. Dana Skorupa, assistant research professor in chemical and biological engineering at Montana State University. Additional support for this podcast also comes from Montana State University. As we wrap up season 2, we want to thank you again for listening to Bison Jam. We're hard at work on season 3, so be on the lookout for new episodes coming in summer 2026. See you soon.